Hello, everyone. My name is Ananth Jagannath, and today my presentation is on the microgravity effect on metamorphic protein structures. Companies such as SpaceX and NASA are planning on proposing to establish future bases on different planets. However, space travel takes a really long time. Just getting to our home planet Mars, or neighboring planet Mars, takes nearly 9 months. So if you want to support life in space, we will need to be able to synthesize and create medical applications that are specifically tailored to the microgravity environments and different gravitational environments in space. Unfortunately, not much research has been conducted on how these gravitational forces affect proteins and protein structure. So today I'm going to propose 2 experiments to establish a way to find out how these gravitational forces influence protein structures. Before I start, I'm going to give a quick overview on protein synthesis. On Earth, most proteins follow this line of function, where they're expressed from mRNAs, and then amino acid chains interact with each other to create a functional protein shape. In 2012, a new class of proteins were discovered. Metamorphic proteins are proteins that have 2 or more conformational states, and environmental factors such as pH and temperature influence which states these metamorphic proteins can be found. As these environmental factors changes, so does the state of the protein and the functions of the protein. We can use metamorphic proteins to determine if gravitational forces are one of these environmental factors that are influencing protein structures. The metamorphic protein I plan on using today is the XCL1 protein. It's in equilibrium under physiological conditions and has 2 states. State 1 is a chemokine structure with alpha-beta folds, and it binds to chemokine receptors. State 2 is a beta-plated sheet and has 4 beta folds and binds to a specific sugar. My hypothesis is that the lack of gravitational forces in space will influence protein synthesis to favor one metamorphic conformation over the other. The first experiment is the ambitious experiment. I'll be using the hydrogen-deuterium exchange to determine how gravitational forces affect the protein's conformation. Deuterium is a heavy isotope of hydrogen with an extra neutron. It forms similar bonds to that of water and is often called heavy water. On the protein, there are many hydrogen atoms represented by the red H's. The hydrogens are located both on the peripheral of the protein and on the interior. When the protein is exposed to the deuterium solution, the hydrogen atoms exchange places with the deuterium. We can then cleave certain sequences of the protein, analyze their hydrogen to deuterium ratio, and determine what protein we are using. Before we start the ambitious experiment, we will need to do a control test to ensure that deuterium behaves similarly on Earth and space. We can use an actin protein, as this protein is a well-studied protein and we know a lot about it. We can allow the hydrogen-deuterium exchange to take place both on Earth and space. And over time, if we get the same ratios of hydrogen-deuterium on the proteins, that means that the gravitational forces are not influencing the hydrogen-deuterium exchange, and we can continue our experiment. To start with, we'll use the BioBits in all 8 of our tubes. The BioBits is a cell-free protein synthesizing tool that expresses DNA to create proteins. Once we insert the XCL1 DNA to the BioBits, in space, the proteins will be synthesized, and the environmental factors will influence state 1 and state 2 conformations. Next, we will add DNases and RNases to prevent further protein synthesis. This is crucial to ensure crucial to add before we add our deuterium, to ensure that as the deuterium exchange takes place, no more proteins are being synthesized, as it could skew our results and shade the true results of the experiment. We would then add the deuterium solution and allow the deuterium exchange to take place. the more peripheral hydrogens are in higher contact with the deuterium solution, so they're more likely to exchange places. The interior hydrogens of the protein are less exposed to the deuterium solution and take longer to exchange places with the deuterium. We then will conduct a similar experiment on Earth and bring all our proteins back to Earth. We can use mass spectrometry in two ways to analyze our results. One way is by examining similar sequences between all our proteins. We can put these sequences through the mass spectrometry, and it will reveal the hydrogen to deuterium ratio of these sequences. If we find any differences, it will indicate that there are different proteins being produced on space and on Earth, and thus gravitational forces are influencing the protein's conformations. Another way we can use mass spectrometry is by compiling all our data on space and all our data on Earth. We can create a graph like this that measures the percent of deuterium throughout the N-terminus and C-terminus of the proteins, and any differences would indicate that different proteins are being produced. And once again, gravitational forces are influencing protein structures. That concludes the ambitious experiments. The next experiment is the freeze and flies experiment. It utilizes fusion proteins and split green fluorescence proteins. A fusion protein is when we have two full proteins that code for two full DNA strands that code for two different proteins. When this is expressed, the fused DNA strands will create one fusion protein with two main domains, and they carry out the same functions as when the proteins are expressed separately from one another. The next is the split green fluorescence protein. A green fluorescence protein is a protein that emits green light when shown under certain light conditions. A split green fluorescence protein, or split GFP, is when we express a part of the DNA strand of the green fluorescence protein to express part of the green fluorescence protein. When two parts come together to create a whole, they create an active green fluorescing protein. Once again, I'll be using the metamorphic protein XCL1, this time taking advantage of the fact that state 1, the chemokine structure, only binds to chemokine receptors. State 2 does not bind to chemokine receptors. To start off with, in 7 of our tubes, we'll have the BioBits, the cell-free protein synthesizing tool, and chemokine receptors attached to 80% green fluorescence protein. We will then add the metamorphic protein DNA fused to 20% of the GFP protein DNA. When synthesized, we will get state 1 and state 2, both with 20% GFP proteins. When state 1, the chemokine structure, binds to the chemokine receptor, it would also induce the GFP proteins to bind together and glow. So when we see glowing, we will know we have lots of state 1. State 2, on the other hand, does not bind to chemokine receptors, so if we see a lot of state 2, we should not see any glowing. We can then use the fluorescence viewer to view our results. One way we can quantify our data is by comparing our metamorphic protein tubes to a fluorescence grade. We can use cyclin-dependent kinases, or CDKs, and cyclins for this part. Cyclin-dependent kinases and cyclins are important steps in cell processes. By keeping the amount of cyclins constant, but varying the amount of CDKs, we can create a fluorescence grade of no fluorescence to high fluorescence. In relations to our metamorphic protein tubes, no fluorescence would correlate to no chemokine structures, and lots of fluorescence would indicate lots of chemokine structures. We can then compare similar amounts of fluorescence to our CDKs to get an approximate ratio of the metamorphic proteins produced. From these experiments, we'll be able to understand how gravitational forces influence protein synthesis, how gravity affects already made protein structures, and through these experiments, it'll allow us to create future improvements in areas of research for drug formulations in space. These projects will enable us to make a safer, much more reliable medical application for space exploration. I'd like to acknowledge my mentor, Mr. Michael Dass, and the Genes of Space team for making this presentation possible. Thank you.